What's up, biology students? Mr. Holloway here. In today's video, we're going to talk about how our genes are regulated and the kinds of factors that cause certain genes to turn off while others get turned on. Humans have about 24,000 protein coding genes, but they are not all being used to make proteins all at the same time. Many of our genes turn on and off as needed and are expressed only when conditions are right. When a gene is expressed, that means it is turned on and is actively being used to build proteins. And gene regulation within the cell helps to control which of our genes are being expressed at any given time. Our genes hold information that the cell can use to assemble protein molecules. According to the central dogma, genetic information in the cell flows from DNA to RNA to proteins, and these proteins perform a huge variety of functions inside our cells, functions which ultimately give us our traits. Essentially, this diagram here is showing us what is happening when a gene is being expressed by the cell. Gene expression is important for us to understand because differences in gene expression account for all of the differences between all of the different kinds of cells that make up the organs and tissues in our body. For the most part, every single one of our cells has the same DNA inside, and every cell in our body contains the same set of approximately 24,000 genes. But the way that these genes are expressed varies from cell to cell. Although many of the genes expressed in our cells are the same no matter what kind of cell we're talking about, they are all human cells after all, there are also notable differences in gene expression from cell to cell, and these differences explain why our nerve cells are different in structure and function than our muscle cells, even though they are both human cells that make up human organisms. Different genes are expressed in a nerve cell than in a muscle cell, and some genes that are turned off in a nerve cell are turned on in a muscle cell. This means that muscle cells and nerve cells contain a different set of proteins to perform a different set of functions. Likewise, bone cells and blood cells have different genes expressed inside of them as well, and genes that are turned off in one kind of cell may be turned on in the other. The same is true of any organism. All frogs, for example, have fundamentally similar DNA. That's what makes them frogs and not something else. But within a frog, different frog genes are expressed in different frog cells, and that helps these cells to specialize and take on specific functions, just like our nerve cells, muscle cells, bone cells, and blood cells have specific functions in our bodies. But how do our cells know which genes to express and which to turn off? Very often, genes are turned on or off in response to environmental or chemical cues, stuff in the environment that our bodies respond to. This process is a bit different depending on whether we're talking about prokaryotes or eukaryotes. In eukaryotes, for example, every gene is regulated individually, which means that the construction of each and every one of our proteins is also regulated individually. In prokaryotes, single-celled organisms that lack membrane-bound organelles, groups of genes are often regulated together, and these groups of genes are called operons. In prokaryotes like the E. coli bacteria depicted here, operons like the Lac Operon are responsible for manufacturing several related protein molecules, and the manufacture of all of these proteins is controlled by the same regulatory mechanism. The Lac Operon has been studied in detail, and is an excellent example of how gene regulation works inside the cells of living organisms. The Lac Operon contains genes necessary for metabolizing the sugar lactose, which is how this operon gets its name. These genes code for a set of enzymes that help to metabolize lactose in E. coli bacteria, and these genes are only expressed when lactose is present. That way, the bacterium is not manufacturing proteins that it doesn't need and cannot use, because that would be a waste of energy and resources. When lactose is absent, as is the case here, this whole set of genes is turned off. Here's how that works. In this figure, this region referred to as the regulator gene is responsible for producing a protein that represses or prevents the transcription of the genes necessary to metabolize lactose. When this repressor protein is active, which it is anytime lactose is absent from the cell, it binds to this region of DNA called an operator. By doing so, this repressor protein essentially acts as a roadblock and prevents RNA polymerase from being able to bind to the promoter region here, which means that RNA polymerase is unable to transcribe these genes. Effectively, this operon is turned off and these genes are not being expressed. However, when lactose is present, the repressor protein becomes inactive. That's because lactose binds to the repressor, causing it to change shape. Shape is very important to a protein because the shape of a protein is what allows that protein to do its job. So when lactose binds to this repressor and causes it to change shape, the repressor is no longer able to bind to the operator region. With nothing standing in its way, RNA polymerase is able to attach itself to the promoter region and is able to transcribe this DNA into a sequence of mRNA. That mRNA can be used by the cell to make the enzymes necessary to metabolize lactose. Then, when all the lactose is metabolized and gone, the repressors become active again, binding to the operator region and preventing any further transcription of DNA. This shuts down the production of proteins until the next time lactose is present. For the most part, gene expression in eukaryotes is regulated in much the same way. Genes are turned on or turned off in response to the presence or absence of some chemical or an environmental factor. The major difference between prokaryotes and eukaryotes is that in eukaryotes, every gene is regulated individually. No operons, no groups of genes being regulated together. And in many cases, more regulatory protein molecules are involved. These regulatory proteins, called transcription factors, help enzymes like RNA polymerase to access the piece of DNA that needs to be transcribed. In this figure here, all these transcription factors here are binding to a region of DNA called the enhancer region, forming a large protein complex made of many individual protein molecules. This protein complex makes it easier for RNA polymerase enzymes to bind to the promoter region in order to begin transcription. There are a few other factors depicted here that also contribute to the regulation of gene expression, including the presence or absence of these methyl groups or binding proteins that bind to specific sequences of DNA. And both of these things, both methyl groups and binding proteins, can get in the way and prevent transcription of a gene, essentially turning that gene back off again. In humans, during adolescence, the production of certain hormones like testosterone and estrogen signals certain genes to become active and others to get turned off. And the change in gene expression that results from the increased production of these hormones means that a different set of proteins will be produced. The function of these proteins ultimately results in the development of our secondary sexual characteristics and causes our shift from children to sexually mature adults. Both women and men start to grow hair in new places. Women begin menstruation, grow breasts and wider hips, while men develop a deeper voice, facial hair, and broader shoulders, all brought about by changes in gene expression due to changes in the concentration of hormones inside of our bodies. We look, think, and act differently as a result of this process. The changes that occur to our bodies during puberty are pretty big and life-changing, and are also frequently awkward and uncomfortable. But these changes ultimately allow us to mature and to grow up. And it all has to do with gene expression and the regulatory mechanisms that cause certain genes to get turned on while other genes get turned off. In a big way, genes make us who we are. But it is the way that our genes are expressed that really controls how our bodies develop and change over time. Gene expression is what makes our skin cells different than our muscle cells, even though both kinds of cells contain pretty much exactly the same set of genes overall. Understanding how the expression of genes is regulated helps us to better understand why we are the way we are and how we have come to be that way. And with that, I will bring our video to a close. Thanks for watching, and feel free to go back and watch this video as many times as you need until you feel like you understand how the expression of our genes is regulated inside of our cells.